Hi everyone, welcome to a new session of the HPP Innovation Week. I am Margarida Rodrigues, HPP Applications Specialist at Hiperbaric, Spain. In the next few minutes, we are going to enjoy an interesting session titled Principles and Effects, Microbiology Beyond HPP with my colleagues, Dr. Mario Gonzalez and Dr. Vinicio Sermons. Hi Mario, hi Vinicio. Hi.
Yeah.
Hi Margarida.
Between the three of us, we will let you know how bacteria, viruses, and parasites behave under pressure. We will start with Mario. He will talk about bacteria, and then we are going to talk about viruses with Vinicio, and then I will be centered in parasites. Before we kick off the session, I am going to briefly introduce my colleague Mario. So Mario has been working with Hiperbaric Spain for the last 6 years. He has recently obtained his PhD, with his research activities focused on the validation of HPP technology as a safe approach for preservation of low-acid beverages, such as coconut water. Mario is enrolled in several activities at the company, being one of them the validation of our bulk equipment. Before we start, I would just like to remember you that we have an open chat where you can just send us your questions and we will answer them in the upcoming days, okay? So Mario, when you're ready, you can start by sharing your screen. I will confirm you.
Yeah, is it ready now?
Yes, everything's perfect.
Okay. So hi everyone, thank you for joining this session. And as Margarita said in her nice introduction to me, I would start with this presentation regarding control of bacteria by high pressure processing in the food industry. I would like to start with some fundamentals. And yeah, maybe the most important thing to keep in mind is that bacteria in food are very diverse. As diverse actually as the three main characters of this movie you have here. We can classify bacteria as the good, which we consider probiotic and beneficial for human health. Also the bad, of course, those are pathogens that pose a risk to human health and require control. But we also have the ugly. They are not necessarily a hazard, but they are responsible of food spoilage. HPP has an effect on this type of bacteria. In the particular case of the good, we have probiotic spores that will survive HPP. This is something positive, something that we want to keep the good bacteria alive. But on the other hand, HPP completely inactivates molds and yeasts. But some pressure-resistant lactic acid bacteria, for instance, remain alive, and this is also positive. We have the bad also, and we are lucky because HPP inactivates vegetative pathogens to safe levels. Unfortunately, pathogenic bacterial spores will survive HPP, so we need additional controls, as I will show later. And regarding the ugly, we are also lucky because HPP completely inactivates molds and yeasts, as I said earlier, and many spoilage species, but some of them are quite resistant to HPP, and we also need additional controls to keep them in safe levels to prevent spoilage. So how does HPP work? Well, here I show the three main pathogens of food concern: E. coli, Listeria monocytogenes, and Salmonella enterica. You have a nice picture of them before HPP. And here in the bottom, I show the effect of HPP on the structure. In all cases, HPP targets different structures, such as the cell membrane or the flagella, or many different structures. And these affections lead to cell death. Okay. Now I try to show more in detail how HPP affects the vital structures of bacteria. This is an example on E. coli, but yeah, other bacteria follow the same ways. For instance, on top we have a healthy cell, and on the bottom we have a cell that has suffered an HPP treatment. So yeah, for instance, HPP dissociates ribosomes, which are the structures that create proteins, so protein synthesis is inhibited. Also, DNA synthesis and expression of proteins is affected, and also the stress response of bacteria is activated, and this requires a lot of energy. So bacteria will die because of this high requirement of energy. Another mechanism is that HPP induces oxidative stress, and yeah, also create some reactive oxygen species to very low levels, but high enough to kill bacteria. And finally, HPP denatures intracellular proteins and enzymes, which affects, of course, metabolism. This slide here tries to show how HPP affects different organisms based on their complexity. For instance, the most pressure-sensitive organisms are plants, vertebrates, and invertebrates, which are very complex and easily inactivated by pressure. Then we have parasites, which are a little bit more complex, but also inactivated at relatively low pressure. My colleague Margarita will later give a nice talk on parasites. Then we find molds and yeasts, which are also quite sensitive to HPP, but more resistant than parasites. And then we face viruses and bacteria, which can also be inactivated at relatively low pressures. But we have some examples of bacteria that are also are able to survive HPP, even at intense conditions, such as 6,000 bar. Yeah, my colleague Vinicio will focus on viruses later. And on top, we have bacterial spores, which are highly resistant, and we cannot actually inactivate them by HPP, so we need additional controls. Regarding molds and yeasts, those are eukaryotic cells, which are very complex, so it's quite easy to damage them with pressure. On the right, you have the different structures that are typical of eukaryotic cells that are affected by HPP. So the disruption of these structures leads to cell death. They are typically inactivated between 2,500 bar and 3,500 bar. Ascospores are quite pressure resistant, more than vegetative fungi cells, but they can be inactivated at higher pressures as well. Regarding bacteria, those belong to the prokaryotic kingdom of cells, so they are more simple and they can survive HPP more easily. They are typically inactivated between 3,500 bar to 6,000 bar, but we have some species that survive those high levels. Gram-positive bacteria is generally more resistant than gram-negative. For instance, Listeria is usually more resistant than some Salmonella species, and this is explained partially because of the peptidoglycan layer that is thicker in gram-positive bacteria. And bacterial spores are not inactivated by HPP, so we need additional controls. Regarding these spores, we can think about them as dormant structures formed by some bacteria. Not all species, but only some are able to create spores. And as I said, they are not inactivated by typical HPP parameters. You can see in the screen the typical parameters of industrial equipment. The good thing is that most of them are not dangerous while dormant, so control measures must be implemented to avoid this germination step that you can see at the bottom. So some control measures that prevent spore germination are temperature, low pH, and low water activity, and of course chemical additives. And more favorable conditions such as high temperature, high pH, or high water activity will enhance germination, and that's what we don't want. The inactivation of spores can be achieved combining high pressure with high temperature. We will have a nice session next Wednesday dealing with this strategy to control bacterial spores. Good thing as well is that bacterial probiotic spores are able to survive HPP, so they can be used in the formulation of innovative products. Here I have two examples from Ganeren and Christian Hansen that they provide these Bacillus coagulant spores that can be used to develop nice products, such as the ones we have here from Suja. Okay, so can bacteria be controlled by HPP? Well, before answering this question, we have to think about two parameters, the process parameters and the product parameters. Regarding process parameters, we have the target pressure and the holding time during which we held this pressure. And regarding product parameters, we have to think about pH and water activity. This slide shows that low pH values and high water activity values act in synergy in activating bacteria. The graph on the left shows that moving to lower pH values increases the inactivation of Listeria, E. coli, and Salmonella at a given pressure. On the right, we can see on the other hand that reducing water activity also reduces the inactivation we have for Listeria monocytogenes, in this case, at a given pressure as well. So process validation should take into account these physicochemical parameters, not only the process parameters, but also the product parameters. This slide here shows the many different applications of HPP technologies. This means we have commercial products in all of these sectors. But each application has some pertinent pathogens that should be considered. I'm not going to discuss all of these, but for instance, in seafood, you can see that Vibrio may be of concern, and HPP can, of course, control Vibrio. And the same goes for the different applications, okay? This is summarized here. In the particular case of the US, FDA demands 5-log reduction of pertinent pathogens during the whole shelf life of the products. As you may know, the US is the biggest market for HPP products. This means that all the products you see in the US market have been successfully validated. Similarly, the USDA requires the total inactivation of Listeria monocytogenes in meat products. And this is something we can achieve with HPP. So yeah, again, all cured meat products we see in the US market have no Listeria, and most of them, this is because of HPP, because we can guarantee absence of Listeria with HPP in this type of products. Same requirement goes for Japan and other markets with demanding regulation. And it is also important to mention that HPP inactivates spoilage microorganisms. This drastically extends shelf life of food and helps to reduce food waste. So this is another nice benefit of HPP technology. Well, this was all from my side. I hope you enjoyed my presentation. Now I will give the word to my colleagues, again, Margarita and Vinicio. Thank you.
Thank you very much, Mario, for the interesting presentation. Before I pass the word on to Vinicio, I am going to briefly introduce him. So I'm going to remember everyone that we have a chat, so you can just put your questions below and we will go and answer them in the upcoming days, okay? So before I pass the word on to you, Vinicio, I'm going to say a few words about your journey with Hiperbaric. So Vinicio has more than 11 years of experience in HPP. He has done a PhD focused on the mathematical modeling and statistical analysis of HPP microbial inactivation genetics, and joined Hiperbaric in 2017. He now provides technical advice and performs validation tests in the continent of America. So Vinicio, when you are ready, you can start by sharing your screen. Vinicio will focus in viruses, like I said before. Okay.
Yes.
Perfect.
Okay. Hello, everyone. Thank you, Margarita, for the kind introduction. It is a pleasure to be here and welcome to the session, or the second part of the session. As stated, I will be focusing on HPP and as a way to control foodborne viruses. So foodborne viruses, there's lots of attention for the bacteria, but it is actually viruses, the ones that can have the most incidence in terms of foodborne illnesses. At least in the United States, the norovirus, which is actually having targeted or shared as a perfect human pathogen. So it's very stable in environmental conditions, can survive in the food, in the environment. It needs a it's very infectious, and it needs a very low dose in order to infect humans. So overall, in the United States, according to the Center Disease Control data, there's approximately 20 million people that each year are infected with norovirus. From this, you can see that it's about 100,000 people that require hospitalization. So from the people that get infected, 5 for every 1,000 can get into a hospitalization. It's relatively, or not a very low risk, and 1,000 of them might lead to pass away, to death. So it's not only norovirus, but there's also other viruses such as hepatitis that are common foodborne pathogens. And you can see over here briefly the mechanisms by which the virus are transmitted. So you can get mild symptoms, everything from diarrhea, vomit, which can then get aerosolized and transmitted into the other humans, into the environment. You can see it can contaminate, again, the person-to-person contact is an important part of transmission, but it's actually through food, the one that comprises the largest portions of the cases for norovirus and other viruses. Overall, well, despite their highly infectious rate, particularly norovirus, viruses are relatively simple microorganisms or structures. You can see over here there's basically two large classifications. The naked virus, in which the nucleic acids, the generic materials they use to replicate, are covered with a protein structure. If we actually take this protein structure and wrap it up in an envelope made up of proteins and lipids, that will be the case of the enveloped viruses. So it's very interesting because viruses by itself are not alive. There can be thought as well, mini machines that are only going to try to find a host in order to replicate. So over here, the virus infection starts when these proteins over here found in the surface of the virus serve as an antenna to identify proteins that are similar or target proteins within the host, in example, human cells. Once they have identified, these proteins also serve as anchors, so they are attached on the outside or the surface of the human cells. Once attached, they use the membrane of the human cell as a cloak, and when they are disguised, they can get into the insides of the cells. Over here, well, they are pretty nasty guests. They will take every resources, all the machinery available within the human cells, and start replicating itself until the cell is depleted, until the human cell its death. And well, that's the way it continues to propagate or expand within a system. So viruses are relatively, again, very stable. They can survive freezing, can remain within the food surfaces for several weeks. They're resistant to chemical disinfectants. Thermal processing obviously is a way to inactivate the viruses, but it's not applicable for all sorts of foods. Some are, as you know, very delicate and can lead to losses, important losses in terms of the nutritional and sensory properties of the foods. So ultraviolet light, it's effective, but it's only limited to the surface or to a certain depth within the food. And viruses can survive both radiation and ultrasound in terms of non-thermal processing technologies. HPP overcomes these other technologies with the advantage that pressure transmission is uniform, instantaneous throughout the food and the vessel, so it can lead to very effective virus inactivation. And in fact, there are lots of studies that report that at relatively low pressure levels between the 250 megapascal to 400 megapascal range, it will be around 30,000 psi to the 60,000 psi, you can actually achieve or exceed the 5 log reductions for the virus content. So overall, we see over here on the plots or the images on the right, some of the inactivation mechanisms for virus. So basically with the HPP, you will modify that three-dimensional structure of the proteins, allowing the genetic material to come into contact outside this case or the capsid. And once it's outside of the capsid, well, it's susceptible to breakdown by enzymes such as the RNAs. So each living organism has these sorts of enzymes in order to remove any foreign genetic material, and that's the case in this example for the norovirus. Other viruses, such as the vesicular stomatitis virus for cows, they're a little bit more resistant, but they have similar inactivation mechanisms. You disrupt the envelope, expose the RNA or the genetic materials, and in this case, the RNA is very well protected by another layer of protein, so it can remain stable, but overall it can remain inactivated, the virus. In fact, well, the Food Safety and Preventive Control Alliance over here in the chapter 4, page 18, state that HPP can be an effective control to minimize risk associated with foodborne viruses. And next we have just a few examples of this for murine norovirus, which is a surrogate for the pathogenic norovirus. As you increase the pressure level over here, 350 megapascals, 400 megapascals, you induce a greater reduction. You can see that the amount of reductions varies a little bit depending on the food system, the pH, and other components within the food might affect inactivation. And this is another example over here. We have inactivation in green onions, and strawberry purees. So overall, there's also lots of examples. Again, it's a relatively low pressure levels, the one you need to eliminate or minimize the risk associated with foodborne viruses. And now we jump into a hot topic, the COVID-19. First, just a disclaimer that COVID-19, it's not a foodborne virus. It's transmitted through aerosols or through the respiratory tract in humans. So even though, well, there's been lots of questions, lots of inquiries, but overall, COVID-19 is not transmitted through the food or the packaging. Up to date, well, there are not any studies evaluating the inactivation effects of HPP on COVID-19, but there are reports that evaluate this on similar viruses, just such as the avian influenza virus. You can see over here in the plots on the right that 500 megapascals, which would be the black diamonds over here, are sufficient to exceed 5 log reductions both in lab media or in chicken meat. Over here, it's relatively short holding times as well. Nonetheless, there's recent very interesting studies in which they have evaluated human breast milk from patients that have gotten the COVID. They have observed and determined that the mothers can actually generate antibodies that protect the babies from COVID-19 after they get infected. Over here we see the forest study in 29 volunteers that were confirmed with infection, generated these antibodies against COVID. Nonetheless, well, human milk can still be hazardous if it's extracted through pumps. If you do not have the adequate hygienic procedures, it can lead to foodborne illnesses. And in this case, we have newborn babies that have not quite developed their immune system. So color pasteurization, over here you can see 63 or 62.5 Celsius, 30 minutes, reduces the risk with foodborne pathogens, but as we know, it can take away some of the important components present in human breast milk. And in this case, HPP, there's quite a lot of references that say that has a better retention of these bioactive compounds of interest. And in case that you are curious about it, we can provide a white paper on this topic. But carrying over with the study I was mentioning, the one that evaluated the presence of antibodies that gain COVID-19 in human breast milk, you can see over here the comparison in terms of the content for antibody immunoglobulin A. With HPP, you got a greater retention. Another antibody, when compared to thermal pasteurization, another antibody that has targets specifically a part of the COVID-19 structure, the S protein. You can see that there's barely a slight difference, but it's overall the same content before and after the HPP when compared to the Holder pasteurization treatment. Well, it's not only about the content, but actually how effective can the remaining immunoglobulins act against COVID-19. In this case, they performed two studies. The first one, it was against a pseudovirus of COVID-19. You can see the unpasteurized milk. It's about threefold the dilution over here needed to inactivate or inhibit 50% of the pseudovirus, and that HPP closely follows. So they are approximately the same values, implying that after HPP, this breast milk still contains this replication inhibition activity. And this was not observed for the Holder pasteurization treatment, as you can see clearly over here on the green bar. Another of the trials within the study will actually use the clinical isolate of COVID-19. You can see that, well, in this case, both unpasteurized milk and HPP samples were the ones that demonstrated the prevention of infection regarding COVID-19. Basically the same percentages before and after, in this case, the untreated and the HPP. So overall, it's a very interesting application again, and the authors concluded that HPP will become a more suitable method or intervention to yield a safe human breast milk due to the retention of the immunoglobulins, and most importantly, the activity not only neutralizing, but also the propagation, but also reducing the rate of infection with COVID-19 clinical isolates. So as of now, this has been the end of my presentation, and I will, if you have any questions, you can contact me at apps@hiperbaric.com, and I will hand over the reins of the presentation to Margarita. Thank you for your attention.
Thank you so much, Vinicio, for the great presentation. Before I start my presentation, I am going to also briefly introduce myself. So my journey with HPP started in my master thesis when I worked with HPP as an extraction technique to extract bioactive compounds from microalgae to enrich fruit juices. And for the past year and a half, I've been working along with my colleagues in the applications department here at Hiperbaric, giving support to clients all over the world. So I am now going to start by sharing my screen. Okay. Let me know if you can see it. Vinicio or Mario. Can you see it?
Yes, it's ready.
Okay, thank you. Okay, so like I said before, I will be focusing in parasites. As my colleague Mario said before, we have seen that the organisms have different sensitive to pressure. And parasites are usually more sensitive than viruses and then bacteria. And we see here that they are inactivated around 300 to 500 megapascal. And like Mario said before, usually the eukaryotic microorganisms are more sensitive than the prokaryotic ones, because the higher the complexity of the organism, the greater is its sensitivity to pressure. And actually, several parasites have been transmitted through food and water, and they have been the cause of foodborne or waterborne illness. And they range in size, going from one-cell organisms like Cryptosporidium parvum, called the protozoa, to large and multicellular organisms, very complex, like Anisakis simplex, where we can see them by the naked eye. These parasites, like I said before, can be transmitted through food. They can be transmitted by meat, which is an example of Trichinella and Toxoplasma gondii, usually by pork, by fish, by crustaceans, like the example of Anisakis simplex, also by fruits and vegetables and water, as we have the example of Cryptosporidium parvum. Throughout my presentation, I will be focusing in several case studies, and because there are the literature in this matter is very scarce, but I will try to let you know the potential of this technology in inactivating parasites. Okay, so I'm going to start with Trichinella. Trichinella leads to a worldwide foodborne parasitic disease known as trichinellosis that is caused by eating raw or undercooked meat, usually pork and its products, that are infected with these larvae. Trichinella has a wide range of hosts and can infect more than 150 animals, including humans. So I'm going to go to the first case study where the authors studied if HPP could inactivate Trichinella spiralis in Genoa salami, which is usually made by pork and cannot be subjected to heat because it will affect its sensory properties. And what the processors usually do is that they apply curing in order to inactivate this pathogen. And in this case, the authors wanted to include HPP to see if it was suitable to avoid fermentation and drying. However, with the fermentation and drying, they obtained a 2.3 log of inactivation of the larvae, which was all the load. And when they apply HPP, it was confirmed by Moss bioassay. And by apply HPP, since they didn't have any more parasites to inactivate, they actually obtained a very nice inactivation of 5 logs of Listeria and 5 logs of E. coli and 3 logs of Salmonella by using 600 megapascal for 5 minutes with HPP. And to see the effect of HPP in these parasites, they harvest masseter muscle from pig, which was contaminated with Trichinella, and they applied HPP. By using 483 megapascal for 1 minute, they were able to obtain 3.2 logi inactivation. Therefore, they concluded that HPP could be an effective method by an alternative to curing in fermented meats. Okay, now we are going to go to Toxoplasma gondii, which can cause the disease of toxoplasmosis. Is a protozoan. The Toxoplasma gondii is a protozoan parasite that infects most species of warm-blooded animals, including humans, and causes the disease, like I said before, of toxoplasmosis. It can be it can be passed when people eat contaminated meat of animals harboring tissue cysts, and even from the mother to the fetus, and when people consume food or water contaminated with these parasites. I'm going to go to a new study in this case. So here we have ground pork infected meat with Toxoplasma gondii tissue cysts, and it will apply pressure from 100 to 400 megapascal during 30 to 90 seconds. And we can see the results here. So they tested this by a bioassay in mouse, and we can see here that when we apply a pressure superior than 300 for more than 90 seconds, we don't have positive mice. So it was enough that HPP could be used to render meat products free of viable Toxoplasma gondii tissue cysts. And when you go to another study, in this case, Toxoplasma gondii oocytes, not cysts, but oocytes, which are in general more resistant, were inoculated in raspberries in an order of 10 to the 4th. And when they apply 100 megapascal to 500 megapascal for 60 seconds, we see here the results. It was needed a pressure above 300, in this case 340 for 60 seconds, in order to inactivate the parasite in raspberries. And now I'm going to go to Anisakis. Anisakis causes the parasitic disease of Anisakiasis, like I said before in this case. That can invade the stomach wall or intestine of humans. The transmission of this disease occurs when infective larvae are ingested from fish or squid undercooked. In this case, we can see here the Anisakis simplex larvae in salmon, which is very typical, unfortunately. Okay, so now here I have a literature review with different case studies about the minimal HPP conditions needed to achieve no parasite infectivity. And we can see here, we have different food systems, Nile perch fillet, mackerel fillet, halibut, and arrowtooth flounder. And we can see here that the pressure levels range from 200 to 400 megapascal. And we can use, when we are working in a higher pressure, we can have a processing time of around 1 minute. In this case, I'm going to talk also about a side effect that can come when we apply HPP to raw fish, and that is usually the effect on color, because when we apply a very high pressure, usually above 500 megapascal, it is possible to extend the shelf life because we inactivate pathogens. However, it comes associated with protein denaturation, like the denaturation of myoglobin, and leads to this effect on fish. But if you would like to know more, I will invite you to see our session on Wednesday about HPP for fish products, because this will depend on the species of the fish. Okay, so now jumping on to Cryptosporidium parvum, which is actually a very well-known parasite. It has an ubiquitous occurrence in the environment. It is transmitted via oocytes and is shed in feces. We see that it can be found in water, soil, food, and surfaces, and it is a microorganism of concern by the FDA that demands a 5-log inactivation of this pathogen in apple juice and apple cider throughout the shelf life of this product. So this is a particular case of the US, of course. Okay, so now here I have another case study where Cryptosporidium parvum oocytes were inoculated in apple and orange juice. And when we apply these conditions of 500 megapascal for 30 to 120 seconds, we can see here the results. With only 30 seconds, it's possible to see that we do not have infected mice. But in the case of the apple juice, it was needed 60 seconds to reach this result. With this article, it actually suggests the potential to use HPP in order to achieve a 5-log inactivation of Cryptosporidium parvum oocytes in apple and orange juice. Okay, so now jumping to another study. Here we have Cryptosporidium parvum also, but in oysters, which it was found that there are a lot of oysters and clams contaminated with these parasites, usually in US and in Canada. And so these authors wanted to study the effects of HPP in oysters in order to inactivate this parasite. So here we have a highly contaminated tank with 10 to the 7th oocytes of Cryptosporidium parvum, and it was applied pressure from 305 megapascal to 550. between 60 seconds and 180. And then they performed a bioassay in mice. They could see here, among all the pressure conditions that they tested, only applying 550 megapascal for 3 minutes, they obtained only 2 positive mice with the Cryptosporidium parvum, which means that it will prove to be effective in an activation of this parasite, leading to a 93 reduction of the numbers of infected mice. So I did a very brief presentation because actually we don't have much literature in this matter. But I hope with this presentation it becomes clear the potential that HPP has in order to control foodborne and waterborne parasites. And if you have any questions, like my colleague said before, please don't hesitate to write us to apps, and we will be glad to answer you. Thank you very much. Now going to thank you. I'm going to thank Vinicio and Mario for joining us in this session. Thank you very much. And if you have any questions, please put them on the chat and we will glad answer to them.
Pleasure. Thank you for attending.
Thank you, everyone.
Bye.
Bye-bye.